Thank you for the introduction. It's a privilege to be here. Very nice venue. Big thank you for the organizers, especially Benevolent AI. Great conference, great meeting, excellent speakers this morning. I was having a little emotional moment when I was sitting there and listening to Winston and Mike and Philippe talk, and I felt at home. I feel that finally we are talking about same things, and you will see why. And it's really exciting to be in this field right now, in this age, this year. It seems like AI is taking over life sciences, and we see more and more meetings coming up, and a lot more people are getting involved. So if I can advance, I'll talk a little bit about how we use AI in biopharma R&D. And the company that I'm with is BERG. I'm not going to go and read the slide here. I'm the Chief Analytics Officer in the company, been with the company for many years. To point out a few things, there's a very interesting trend that is happening. I feel today that BERG is more of a conventional, mainstream, kind of boring AI company in life sciences, right? 10, 5, 3 years ago, we were anything but boring. We were practically outliers, as well as Benevolent AI in this field, trying to do something innovative, something completely unconventional in the space that is very conservative. And we're talking about medicine, we're talking drug development. What's happened here, it become possible only because of the way that we were funded. And I need to acknowledge Carl Berg and his vision and his confidence in our direction, in the approach that we were taking in drug discovery and building this pharmaceutical company. We're still angel funded. He's been funding us for many years, and it served the only way that this has happened in the industry, that we have this company build a platform. We've developed multiple programs in drug discovery and diagnostics, and you'll see some of the results. It would be very difficult to do it under any other circumstances. So 3 chapters in my presentation, talk a little bit about the philosophy, and then a couple of examples if I have time. I'll go through one of them most definitely, and then we'll see if I have time for the second one. So what is important here, and what is the idea behind BERG? The original idea was to change the drug discovery process. And we sort of approached it from a completely different angle 9, 10 years ago by sort of focusing on data-driven research. As we realized that a lot of the research organizations, teams, pharmaceutical companies are sort of working in the same neighborhood of molecular entities, looking at same compounds, looking at same leads, and kind of sort of using a little bit of Me Too approach. So what we decided to do is to, so to speak, start from scratch and look at biological models of various diseases. And specifically, we focused on cancer, neurology, and diabetes. So this interrogative biology platform that we worked on for a number of years was built to really take a deeper look at disease pathways and attempt to discover novel things and see what is known versus what we can discover in an unbiased, data-driven way. What's important here is these 4 steps are kind of go in sequence, and it's critical to understand that if we were to build useful and actionable disease models, we have to start from in vitro modeling. It's very difficult, understandably, to do models in humans. It's a little controversial to do models in animals because there are a lot of physiology that is very different. So we work primarily with human cell lines or human cell cultures. And it's interesting that a lot of the work that's been done prior, where we'll look at gene expression data, where we'll look at proteomics and other omics or DNA data, is typically done at some sort of baseline conditions. So if we talk about oncology, you'll find a lot of baseline data for cancer cell lines, and there's maybe 1,500 cell lines, and you see gene expression data at baseline. Well, from our perspective, it's not very interesting because it doesn't tell you anything what these cells and their molecular pathways do when they're actually in vivo in a human body, when they compete with immune cells, when they compete with other cells that are present in that microenvironment, where there's maybe hypoxic conditions or there's maybe lack of nutrients, and maybe they're going through anaerobic metabolism. So what we realized that we needed to do is to create disease models, and what's more important, interrogate these models, create perturbations that mimic physiological conditions. And that's what we do in this first stage here. And we create not just a few experimental conditions. We build hundreds and oftentimes thousands of perturbations that attempt in certain way to be sort of intelligently guide us towards certain molecular pathways, but in some aspects just create this sort of random perturbation environment where we can measure the signal at various molecular components of these networks or physiological processes and attempt to understand it by using computational tools. So step number 2, once we build experimental design and we do disease modeling, then we need to measure what's going on as far as the molecular landscape of these cells. And again, we go beyond genomics. And I think Winston had a great comment, is that it's time for us to realize that genomics and next-gen sequencing is not going to solve everything. We actually have to look at biochemistry, because most of the work and most of the activity inside us is done by proteins, enzymes, metabolites, and lipids. And we focus quite a bit on that. We do a lot of proteomics work, and not just total proteomics. We looked at enzymatic activity, we look at phosphorylation, we look at post-translational modifications, we look at the lipidome and the metabolome, as we believe the signal that we're getting from those high-throughput measurements may be even more important than what we can get from genomics. And we do genomics as well with mRNA and DNA sequencing. Now, when we collect these types of data, combined with potentially low-throughput data, such as phenotypic assays or cell proliferation or what other assays that may be relevant for that specific disease, as you can understand, this is huge amounts of data. And it's difficult to analyze these data types by using conventional statistics. We're always getting stuck in multiple testing corrections and the curse of false discovery. We needed new methodologies to take a deeper look and attempt to understand these systems at a more mechanistic level. So, long time ago, we decided to divert our attention from, I would say, more of a classical statistic, machine learning methods that look for associations or correlations, to a method that helped us really create these mechanistic inferences, and it's based on Bayesian Artificial Intelligence. So what we do here, we infer or optimize Bayesian networks, and we work with these disease models networks that give us certain idea of what is happening in the disease environment, as Winston as well mentioned that, and compare it to what is happening in normal environment, and really analyze the topological differences between the two sort of outcomes, two environments. And then on top of that, as some of these networks can be extremely intimidating, they're huge, as you can imagine. There's thousands of features in these networks and very densely connected. We've developed automatic algorithms to really rank order our leads and identify a few, maybe 24 top molecular entities that we want to pursue in our validation programs as potential therapeutic targets or biomarkers. And what's important to notice here is that, in addition to building this workflow, we have resources and I guess commitment to do laboratory validation of our findings, and this is critical. Whenever we create a program and we go through these steps and we identify potential leads, what happens next is that all those leads are going back into laboratory for in vitro validation, where we can convince ourselves that what we're finding makes sense, what we're finding is real. We see sort of some of the outcomes that have been predicted in the model. And a few words about artificial intelligence and sort of taking this, what we do at a high level. What does it mean? So we are in AI meeting here, AI Bioscience Symposium, and we talk about machine learning, we talk about artificial intelligence. What is the difference? I mean, by definition, AI is something that some sort of intelligence exhibited by machines or software. I mean, we're talking more about software. We're not building self-driving cars here. We're not reproducing Siri or anything like that. It's software that allows us to learn, that allows us to engineer knowledge from data, and maybe we can approach reasoning in the near future. And what I mean by that, can this software reason well enough to identify therapeutic targets, to identify leads for us, so that it's not specific scientist or a group of scientists deciding that this compound needs to be targeted with a small molecule. Can this software or artificial intelligence decide this for us? And can we create an automated workflow? And I think it's possible. So when we talk about artificial intelligence and scientific research, what effectively we're trying to do here is automate this workflow of research in life sciences. What you see on the left is a very simple diagram from Bayesian networks, and we as humans can understand it very quickly, right? We have three features here. We have sprinkler, yes, no, rain, the grass is getting wet. We can deduce and reason that if it rains, the grass is going to be wet. If we turn the sprinkler on, it's wet. If we have a sensor for rain, then rain is going to be inhibiting sprinkler, and that's not going to come off, but the grass is going to be wet anyways. So we can figure it out, right? What you see on the right is scientific intelligence. This is years and years of research that we were able to figure out basic glycolytic pathway and step by step understand the enzymatic reactions in the pathway, the substrates, and the products. And the same with the Krebs cycle, oxidative phosphorylation. We understand it really well, and it's great. It's taking us probably, 30, 40 years to figure it out. Again, these pathways are ever present in every mammalian cell. So it's easy to study them. Winston, I think, said, we need to, I'm going to keep referring because you've covered a lot of the material that I want to cover. But it's important to be able to learn context-specific pathways. Because when we go into brain diseases like Alzheimer's and Parkinson's, we're talking about neuronal cells. We're talking about cells that we haven't studied much. So things that are going on there at the molecular level are not well classified and annotated by this basic context-free research. So how do we do that? We can spend another 40 years and take every cell type and learn these pathways one by one, knocking out genes and looking at what happens. Or we can approach in a different way by doing this type of data-driven discovery and using top-down methodology, statistical methods, such as Bayesian Artificial Intelligence, that allows us to learn these pathways in a very automated way. Is this going to be perfect? Is every connection accurate? No. But we hope that a large majority of these connections are representative of what is happening in these cells. And other benefits of Bayesian networks, these tools are data type and size agnostic. We can literally work with thousands of features and millions of observations. It doesn't happen that often in molecular data, but sometimes we have data sets like that. We circumvent the curse of multiple testing. because we're not doing millions of tests, we're actually building one or a few optimal Bayesian networks. We don't have to deal with FDR in this particular problem. And what's important for drug discovery is that Bayesian networks are able to infer cause and effect relationships. It's sort of taking data analysis to the next level, where we move away from associations when we're not just interested in correlations, but we actually attempt to learn how things work, what is driving what in a specific disease model. And it's very important for drug discovery. Maybe not so important for biomarkers, as we're looking for correlates. But for drug discovery, it's clear that we would like to treat the cause of the disease, not a symptom. And it's an important point here. So this is sort of philosophy and the landscape of our tools and methodologies that we've been using for a number of years. And I'll go through a couple of examples if I have time. The first one is an example from research where we look to identify novel therapeutic targets, and it was in Parkinson's disease, very similar to Alzheimer's. A difficult area to study. Not much is known as far as molecular pathways and how things work. So and this program has been in the works for a few years, and it was actually just recently announced that we are collaborating with AstraZeneca to take this program forward into clinical development. What we've done here is looked at human fibroblast cultures from Parkinson's patients and age-matched normal individuals. We've done very extensive experimental work by doing perturbations of fibroblasts, by inducing molecular signaling and looking at a few molecular outputs: proteomics, phosphoproteomics, metabolomics, and some of the SNP work. Then we've created these data-driven, no prior knowledge, no literature, just based on data variability, Bayesian networks that allow us to look at how these variables, molecular variables, are connected in the disease and normal patients. So if I can advance this. On top of that, when we begin to look at the differences, topological differences, and analyze regulatory mechanisms of these networks, as I said, we summarize it in a very concise rank list of ordered targets. And what I used to do, and this is the most amazing thing, but it's not that amazing anymore because I don't need to prove to you that this is a reasonable approach. But before, 5, 3, 10 years ago, when we would go to this stage and we talk to a number of companies in Cambridge, Massachusetts, we go to the stage, we describe the platform, and then they pretty much show us the door. Because no one would believe that you can actually do something meaningful with AI and data, and not even looking at next-gen sequencing. How could you, right? But we had to convince people that what we do makes sense. And then when you go through this automated process and you get a list of potential therapeutic targets, and in this particular case, I don't know if we got lucky or we're just so good, but the first target on the list was glucosylceramidase for Parkinson's disease. And if you're familiar with the disease, it's a well-known, biggest genetic marker for Parkinson's. So the deficiency in this enzyme causes Gaucher disease, but people that are carriers for Gaucher disease or have only one chromosome or one of the alleles deficient, they are susceptible to develop Parkinson's disease much earlier in their lives, in their 40s. So there's no question that GBA is linked to Parkinson's. It's probably having some kind of causative effect in the disease. What's interesting here is that we're looking at the enzyme itself. This is not genetic feature here. This is the protein, and it's showing up as a potential top target through this analysis. Well, as you might gauge, GBA is well known. It's not something where we'd like to invest more effort and resources into taking it further, but we were interested to see what is linking to GBA in the closest neighborhood. Can we find sort of other molecules that are in the subnetwork, but completely unknown for Parkinson's community, because this is our novel IP. And we've been working on many of these compounds, but one of these that I want to talk about today is PIG3. And this gene is literally, I think it stands for P53 interacting gene number 3. So it tells you pretty much nothing. And that's great, because, hey, now it's our IP, it's our gene, we own it. And we looked at the subnetwork. This is specifically a subnetwork for LRRK2-induced Parkinson's disease for patients, not idiopathic disease, but the LRRK2 disease. And we started sort of doing validations, and I'll walk you through some of this work. Step number 1 was to figure out, all right, we've done this work in fibroblast. Does it translate to brain, to regions that are important in Parkinson's? And this is where public data becomes really useful. We can go to the brain atlas and figure out gene expression for PIG3. And yes, indeed, it's expressed in substantia nigra, which is very important for Parkinson's. And our level of confidence is beginning to grow more and more that this is maybe a real find. So then one of the sort of quick tools that you can use in neurodegeneration is this neuroblastoma cell line. There aren't many in vitro models for neurodegeneration, but SHSY5Y is a well-known model to do some of the initial validation and kind of take your confidence even to the next level, see if this is working. So what we've done here, using this cell line as our in vitro model, we induced some of the damage by chemical compounds, for example, rotenone. It's known to inhibit complex 1, inducing some of the reactive oxygen species response in the cells, and inducing MAP kinase and leading to neurodegeneration. So rotenone is actually, I think, was classified recently by the NIH as some of the compounds that can potentially induce Parkinson's if people are exposed to it. So we used it here to induce neurodegeneration. And as you can see, we see the positive control cleaved PARP, which is a marker for apoptosis. It's downstream of caspase. And we also see that PIG3 protein expression is going up with in the concentration-dependent manner from rotenone exposure. So that tells us sort of next step in the story is that, okay, if we induce neurodegeneration in this in vitro model, we do see induction of the expression of this gene, which is great, confirming that this may be relevant. We use different compound, 6-hydroxydopamine, sort of acting in a similar way at a high level, inducing ROS, leading to rapid neurodegeneration. And again, we see even bigger expression increase in PIG3, and you see positive control over there. So 2 assays, different compounds, different chemical perturbations, looking at the same sort of mechanism and seeing the same effect on our gene of protein of interest. Now, if we go into more of a mechanistic studies, we need to actually act on that gene, right? Because this is what the model predicted, that this thing is important. We need to overexpress or knock it out or knock down. So it's relatively easy to use siRNAs these days, and we've conducted some of these experiments looking at mRNA knockdown. What you see on the left is mRNA knockdown for PIG3, and then what you see on the right is what is happening with that in vitro model when we introduce that siRNA. And what's interesting here is that obviously you see the same induction of expression of PIG3, and you see fold change in cell death for this model. But when you actually knock down that PIG3 by mRNA, you see protective effect happening in these cells. So the red bars on the right at 50-100 micromolar of 6-hydroxydopamine or rotenone, you see significant difference in cell death change, in the fold change. And that's important. Now this is becoming more mechanistic. We're knocking down this molecular entity, and we're seeing interesting phenotypic effect. So to sort of summarize the story, we also looked upstream. We know that MAP kinase is upstream, and it's a sort of well-known mechanism in neurodegeneration. So we needed to figure out where is PIG3 relative to MAP kinase. And what we used here, and again, the same model, is a small molecule that is known to target MAP kinase and inhibit its activity. And what you see again is that if we introduce this small molecule into the culture under the same conditions inducing ROS and neurodegeneration, you see a protective effect of and downregulation of PIG3 mRNA expression with rotenone and 6-hydroxydopamine. So that tells us that PIG3 is downstream of MAP kinase. MAP kinase is really difficult to target because of the other side effects that may happen as this very sort of central gene, central protein for many regulatory mechanisms. But PIG3 may be a better target for Parkinson's, and this is what we're pursuing right now. I think I'm running out of time. I don't know if you guys are going to post the slides. There's the second case study here. If I can move this forward in development. I can't. I wanted to, yeah. So this is more about biomarkers and discovering novel entities for complementary biomarkers in clinical trials and how we use the same system to do so. But I will skip this and we'll just go to the acknowledgement slide where our BERG team and our collaborators from Parkinson's Institute. Thank you.
Thank you very much. We've just got time for 2 questions at most. One over there.
Thanks, really great work. Your target validation on the PIG3 was kind of very detailed and focused on a single gene. If you wanted to do that for sort of higher throughput on multiple targets, do you kind of see a role for things like CRISPR screening or RNAi screening or other higher throughput technologies?
Yeah, that's a good point. I think we want to be a little bit more focused and go deeper rather than spread out across maybe hundreds of potential targets, because in our mind, I think we believe it, what we discover. So PIG3 is really becoming a drug development program where a lot of funding is going to go to.
Okay.
And one last question here.
Oh yeah, Ian Churcher. So I don't know anything about PIG3. It doesn't sound like many people do. How do you assess the tractability of actually getting an agent that you can use in the clinic out of a target like that?
Yeah, so this is focus of our collaboration with AZ is to, PIG3 is a protein that we're targeting. We need to develop a drug to it, and this is what we're doing right now. Sort of screening through multiple technologies, figuring out how we're actually going to be targeting and knocking down that protein in patients.
Okay, I think we're going to have to stop there. The next, thank you very much again.